Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in the course Genetic Engineering Theory and Application, this is our last and final third live sessions. And what I have planned is that in this particular session, you can ask me any questions from this course, and you can. What we are going to do is we are going to summarize the course in some of the slides, and that may actually help you to revise the course content also. And then I will be happy to answer your questions. So before doing so, if you have any question, you can ask me. So it seems that you people don't have any question at this moment. So what I will be going to do is I'm going to share my screen, and then we are actually going to first see what we have discussed or what we have learned in this particular course, and then followed by it may actually revise the content, and at the other end it will help you to ask the queries or confusions, whatever you have. So let me share my screen. And so now can you see this PowerPoint slide, right? And this is the live session 3. And what we are going to do now is we are going to revise the content module by modules. So if you see in the beginning, what we were doing in this course is we were trying to understand how you can be able to, clone a particular gene into the vector of your choice or a suitable vector, right? And then we have also discussed about the how you can actually be able to do a DNA delivery into a suitable host. And then from the host, we have also discussed about the overexpression of the protein. And then followed by we have discussed about the purification. And in the last module, we have also discussed about the characterization. And once you are going to be done with the characterization, this means your product is ready, and then you can actually be able to use that for the application. So we have discussed about the various type of applications of the biotechnology. So if you start, if you recall, we started our journey of this course from the very basic that we have first understood. First we understood what is the what is the basis or basics of the cell biology, right? So in this particular content, we have discussed about the prokaryotic cell. We discussed about the eukaryotic cell. And while we were discussing about the eukaryotic cell, we have also discussed about how you can be able to prepare the different types of mediums for propagating the prokaryotic cell or the eukaryotic cell. We have discussed about, we have even shown you a couple of demos how we can be able to prepare the mammalian cell culture media or the prokaryotic microbiology media, and how you can be able to autoclave and all those kind of things. And then we also discuss about the differences between the prokaryotic and the differences between the prokaryotic and the eukaryotic cell, right? Prokaryotic cells and the eukaryotic cell. So, so initially the idea was to, to clear your basics, and then followed by this, we have also discussed about the isolation of the gene of interest, right? So we have discussed about the genetic genomic library preparation. We have discussed about the cDNA library preparation, and we have also discussed about the PCR. So all these three approaches we have discussed in detail, how you can be able to use them for uh, isolating the gene of interest. And ultimately we have, while we were discussing about the polymerase chain reaction, we discussed about how you can be able to design the primers. So primer designing we have discussed in detail about with the demo also, that we have shown you how you can be able to prepare the primer design, right? And then we have also discussed about the the PCR. So how you can be able to how you can be able to do the PCR. Right? So then we discussed about how you can be able to set up the PCR, right? And for this also we have shown you a demo. And then ultimately we have also discussed about how you can be able to analyze the PCR product, and how you can be able to isolate the gene. Now in the third module, we have discussed about the cloning of the gene into the suitable vector. So in this, we discuss about the vectors, their properties, and then we have also discussed about the different operational stuff related to the cloning of the gene, right? So within this, we have discussed about the restriction enzymes. We have also discussed about the other enzymes which are important for the cloning, right? And once your clone is ready, then in the fourth, we have discussed about the DNA delivery into the host, right? And so within this, we have discussed about the transformation, and we have shown the transformation by the chemical method, right? And we have shown you a demo about the chemical method, and we have shown you that how you can be able to prepare the calcium chloride induced calcium chloride mediated competent cell preparation, and how you can be able to use that for preparing the uh, for transformations. And then we have also discussed about the electroporation, right? And we have shown you a demo about the electroporation as well. Now, once the transformation is done, then the transformation is for the bacterial system. For the mammalian expression system, we discuss about the transfection, and we have also discussed about the bactofaction and so on, right? And we have also discussed about the virus transactions, right? So that all we have discussed, and apart from that, we have also discussed about the liposfectamine mediated uh, transfection. And for some of these, we have also shown you a demo how you can be able to do the transaction and how you can be able to check. So once you are being delivered the clone, right? So DNA is nothing but a clone. Your recombinant organism is ready, and then you can use that for overexpression. So when we were discussing about the overexpression, we have discussed the overexpression in bacterial expression system. We have discussed about the yeast expression system. We have discussed about the mammalian expression system, right? And then we also discussed about the yeast expression system. And from all these three expressions, all the four expression system, we are going to get the protein going to be produced in the large quantity. And then in the uh, subsequent to that, we have also discussed about the sorry. Subsequent to that, we have also discussed about the purification, right? So we have discussed about the different methods of purification. So we have discussed about the chromatography system, and we have shown you a complete demo how you can be able to operate the chromatography system. And then we have also discussed about the ion exchange chromatography. We discussed about the hydrophobic interaction chromatography. We discussed about the gel filtration. And lastly, we have also discussed about the affinity chromatography. And when we were discussing about the affinity chromatography, we discussed about the how you can be able to prepare the column. So in that we have shown you how you can be able to generate the antibody. So we have shown you a small demo about how you can be able to generate the antibody in the rabbits. And then we have also discussed about the nickel NTA affinity column, right? So we have to. And once they're done with the purification, then we have also discussed about the characterization of the product. So when we were discussing about the characterization of the product, we discussed about the how you can be able to even characterize the protein, right? So we can actually do the protein sequencing. And so we have discussed the both method of protein sequencing. And then we have also discussed about the spectroscopic method of, the for characterizing the product. And we have also discussed about the IR spectroscopy and so on. And once the characterization is done, then we have also discussed about the application of the product in context to the biotechnology. So we have discussed about the various types of applications of the product in the field of biotechnology. We have also discussed some of the advancement in the biotechnology where we have discussed about the artificial inseminations and all that. So with this brief discussion about the content what we have discussed so far in this particular course, I think it will be maybe a kind of a uh, revision for you to recall whatever we have discussed. And now I would request you all that you can basically post your questions so that we can be able to have a fruitful discussion, and that may help you, to prepare for your exams. In case you have any more questions related to the uh, exam itself, then you can ask that question as well. And remember that this session is only for 1 hour, so it is a good opportunity for you all to clarify your doubts, and also to to prepare for the forthcoming exams, which is very close. It's now almost 2 weeks apart from now, so it's better to clarify your doubt now. You can also be, post your questions in the discussion forum as well. But since I am available right now. It's better you take this opportunity to ask the questions. So I think there is a question from the Rajat Chaudhary, and he said ion exchange chromatography. So Rajat, can you make it more clear what you want to ask in the ion exchange chromatography? It's better to clarify what you could not understand. Or do you want me to explain the complete ion exchange chromatography? Bharat, can you see, can you hear my voice now? Okay, I think not responding, but I think we'll discuss about the ion exchange chromatography as he asked. So I'm going to share my screen and so that it would be easy for me to discuss, right? And so what he is asking is ion exchange chromatography. Okay. So ion exchange chromatography is a chromatography technique which deals with the charge on the molecule. So it can be either a cation exchange chromatography or it could be a anion exchange chromatography because charge can be of two types, right? So either you can have the positive charge, which you are going to use for exchange, or you can use the negative charge for thing, right? So if you use the cation exchange chromatography, the the protein should be of the positive charge. Okay. And if you are talking about the ion exchange chromatography and ion exchange chromatography, that the protein should have the negative charge. This means, and in this particular ion exchange chromatography, what you're going to do is you're going to have the matrix, and on this, you're going to have the groups, right? So, for example, in the cation exchange chromatography, there are going to be the negative charges. And then for this negative charge, the positively charged protein is actually will go and bind. Okay, so this is going to they have the interactions, right? And in this process, it is actually going to displace. Okay, let me just go a little back, right? So, okay, so in a typical ion exchange chromatography, what you have is you have a matrix, right? So you have the matrix, and on this matrix, for example, in the cation exchange chromatography, you're going to have a negative charge group. Okay, and this negative charge group is going to have the positively charged cation, which is going to bind, right? Now, when you add the protein to this, right? When you add the protein, which is also positively charged, it is actually going to replace this sodium, and as a result, it is actually going to bind to this particular negative charge. Okay, so it's going to bind this negative charge by the protein. Okay, now if you do a washing, what will happen? So after this, you're going to do the washing, right? So the protein is actually going to bind this particular matrix, and this affinity will be proportional to the charge. This means if I have four molecules, one is negatively charged, another one is one positive charge, the second one is two positive charge, and the third one is having the three positive charge, then this one is going to have the highest affinity, This one is going to be in the middle range, right? So if it is highest affinity, I'll give them 1 mark. If it is the middle range, it's going to be 2. And this is going to be third, right? So it's going to be a least affinity. And this one, because it's only also negatively charged, it will not going to have any affinity. This means it's not going to bind the column. So based on these kind of interactions, the protein will bind to a different affinity. Okay. And now what you're going to do is, so when you do a washing, the negatively charged protein or other proteins are actually going to be removed from the matrix. And that's how the matrix is going to have the positively charged protein, which is bound, right? Now what you're going to do is you're going to supply the cation, that is the sodium. And once you supply the sodium, the sodium is actually going to replace this protein. And that's how it is actually going to give you a protein which is negatively bound, and then it also going to have the sodium bound. And in this process, it is actually going to elute the protein which is positively charged. Okay, this displacement is also based on the affinity. Okay, so if this one is going to be displaced at the end, this one is going to be the displaced at the first. Okay, so as per the displacement, this is going to be displaced first, this is going to be displaced second, and this is going to be displaced later third, right? So this is going to be eluted at the end. And that's how all the proteins are going to be separated. In the anion exchange chromatography, exactly the same thing is going to happen, except that the matrix is going to have the positive charge group. So it's going to be exactly the same, but completely reverse. And it's going to have the negatively charged anion which is bound. So in first process, the protein should be negatively charged, right? That's what I said. So protein should be negatively charged. So protein is going to have the negative charge, and that is going to replace the chloride, and that's how it is actually going to bind. And here also exactly the same thing has happened. Okay, that the affinity would be proportional to the charge. So the protein which will have the more number of negative charges will have the highest affinity, and so on. So I hope this by this discussion probably it will be clear to you. Rajat? So I hope that Rajat, it is clear to you now. And if you have any more questions, you can ask me also. Okay, Rajat, I'm happy that it helped you to understand the topic. If you have any more queries, you can ask me. So it seems all your queries are being addressed, and you don't have any more queries. So with this, I would like to conclude the live sessions for the course Genetic Engineering Theory and Application. And I hope you will do good in your endsem exam. And I wish you the best for your exam. So with this, I would like to conclude the session. Thank you.